Okay, I think we can go ahead and start now. And Jen in there. And Josh. Still trying to get you in. So. Okay. Okay, we are going to go ahead and get started. We're talking about the various organelles in the cells. And I'm gonna, we stopped when we were talking about lysosomes. I think it's according to my note where we stopped. Anybody have any questions on anything before we move on? Questions on the test coming up? I think it opened up today. I think it stays open for a week. So if you have any questions, this is a great time to ask. Or you can just shoot me an email, whatever you want to do. Okay. Well, in that case, I'm going to go ahead and go into the PowerPoint and pick up where we stopped the last time. And this was looking at, as I said, lysosomes. Okay, so we're looking at the cell organelles. You know, the organelles are little structures within the cells that do all sorts of things. We looked at the mitochondria and the ribosomes and the endoplasmic reticulum, probably more on that than I wanted to, but it's okay. Talked about the peroxisomes and their ability to destroy free radicals. Now the lysosomes are sacs of digestive enzymes, and they're very dangerous to the cell. So when we make these enzymes, we go ahead and store them inside this sac called a lysosome, so it can't dissolve away the cell proper. Because if these digestive enzymes were to get released into the cell from the lysosome, it would dissolve the cell. In fact, cells will often undergo what we call autolysis or apoptosis to break themselves down when they're damaged beyond repair. But most cells are going to use lysosomes to destroy bacteria, to break down food they've engulfed, to fight infections, to break down other cells that have that are worn out and have already started to break apart. You know, cells that have been infected, cells that have died, dead structures, things like that. Cells use lysosomes for all that. You know, you have organelles that wear out, you replace those and you destroy the old ones. We use them in bone, and we use lysosomes in bone cells to break down bone to release calcium so we can build new bone. And we also use lysosomes to break down glycogen in the liver so that we can release, so we can break it down to glucose. You know, the process of glycogenolysis, the lysing of glycogen molecules into glucose. And so these, we have to keep these enzymes, these digestive enzymes, away from everything else until we're ready to use them. So we keep them in their own vesicles inside the cells, and these vesicles are called lysosomes. Now there is a problem with lysosomes. We use them to control the accumulation of lipids, glycolipids in the brain. And there is a condition known as Tay-Sachs, which if the lysosomes are used to break down the accumulation of glycolipids in the brain. Glycolipids are made as part of the development of the central nervous system. We keep them in check with certain lysosomes. We don't let them grow too big, because if they got too big, they would start compressing brain tissue and destroying brain tissue. And this is all occurring when we are, you know, in infancy. Well, there is a genetic disorder called Tay-Sachs. Tay-Sachs is one of those homozygous recessive disorders where you only have it if both the mother and the father are carriers. And it's a mutation of the DNA that controls the development of these glycolipids. There is no lysosome present that will break down these the excess glycolipids that form. There's a mistake. That's what a mutation is. A mutation is a mistake. And the mistake is that the instructions to make the lysosome enzymes aren't there. The instructions aren't there. And so what will happen is that these glycolipids will accumulate in the brain of the developing child starting in year 1, and usually accumulating enough to destroy the brain by about age 5. And it's not something that you can catch. It's a genetically acquired disorder, and it's limited to a very small population subsets that occur in different locations around the world. It originally was seen in the Jewish populations of Eastern Europe, which was if you know your history, the Jewish populations in Eastern Europe were a very closed society. And when you have a closed society where the population intermarries within itself, not inbreeding, but staying within their own gene pool, these recessive disorders stay alive. Now, what does that have to do with East Tennessee? Well, there's a point to this. So the largest population of individuals with Tay-Sachs disease in their genetic makeup are the Jewish populations in Eastern Europe. However, for whatever luck of the draw with genetics, French Canadians also had this genetic disorder. It show up in their gene pool. French Canadians of several hundred years ago. Now, if you know anything about history, somewhere in the 1700s, 1800s, the French Canadians were living in the area around Nova Scotia and Newfoundland and those areas of Canada. And they were French, and the British took over Canada and kicked the French out, and they went down to Louisiana. And so they were carrying their Tay-Sachs recessive genes with them. And now we have a population subgroup in southern Louisiana. They call themselves the Cajuns, and the Cajuns carry that recessive disorder. And we now have a much closer link to Tennessee, because when Katrina hit and other hurricanes have hit, many people from southern Louisiana have relocated to a little further north away from hurricanes, including East Tennessee. So we have a greater possibility now to start seeing patients who have children with Tay-Sachs. So just something to consider here. What will the symptoms, the primary symptoms you see are, the child usually under the age of 5, because usually they're dead by 5. They have seizures, they have cognitive impairment, they eventually lose their eyesight, their motor coordination, motor skills seem to degrade worse and worse instead of getting better and better as they grow. And there's no treatment for it, and it's going to generally have a sad ending. But there is a greater possibility now to encounter patients with this disorder than there was before. Normally you would expect to only see, people with, that are Tay-Sachs carriers in some of the larger population centers around the nation, particularly where you have a high Jewish population, where they have descended from Eastern European Jews. But we have a fair number of French Canadian descendants that living in Louisiana below New Orleans, and they have migrated, or moved up here to avoid hurricanes. And so we start seeing more and more of them throughout the upper South. So it's just a possibility, something to pay attention to. Okay. Now, Switching gears to something a little more positive, the reason our cells all have distinctive shapes. I mean, consider the fact that a nerve cell is as long as the nerve is, and some nerve cells can be 3 or 4 feet long, and muscle cells are as long as the individual muscles are. So if you have like your calf muscle, it may be 18 inches in length. Well, it's made up of muscle cells that are all 18 inches in length. So the individual cells have the shape of the organ they're in. Why are red cells shaped like a lifesaver? Why are white cells squishy like an amoeba? Why do we have columnar cells and cuboidal cells? Why do we have these distinctive shapes? And it's because these cells have their own type of skeleton, not bony, but made up of filaments and tubules that will hold these cells into their specific shape so they can do their job. They allow the cells to attach to each other. They allow the cells to attach to other structures. They allow the cells to contract thanks to these filaments and microtubules. The cytoskeleton is made up of tubules. First thing to look at are tubules. And these microtubules, we see them in spindle fibers that allow us to move the cells, the cytoplasm and the chromosomes during mitosis. These microtubules, provide movement of different structures within the cell. We also see these tubules being used as the basis for the structure of cilia and flagella, the things that allow motion to occur. The second type of structure are what we call the microfilaments. The microfilaments are the contractile proteins, like actin. Actin is a contractile protein. We see it when we have cytokinesis. It's that band of actin protein wrapping itself around the outside of a cell so that as the daughter cells are forming, cytokinesis can separate the cell, the parent cell, into two distinct cells, two distinct daughter cells. And the third category are the intermediate filaments. For example, that's like keratin, the waterproofing compound in our fingernails and in our hair and our skin. They allow us to be water resistant and have layers of protection. Our skin contains lots of keratin in there, so that yes, it is stratified squamous epithelial cells, but they're keratinized. So not only are they in multiple layers, they're going to be waterproof, water resistant, pathogen resistant, if you will, too. Whereas we have stratified squamous cells in our mouth, they're not keratinized, they're not waterproof. They release lots of mucus to help us eat food, but that mucus also has certain antibacterial properties. Anyway, the point is 3 different types of filament type structures here for the cell, enable the cells to do things. Another structure we encounter inside the cells is the centrioles. Now, the centrioles are little barrel-shaped structures. They are made up with the same arrangement using those microtubules that we see with cilia and flagella. And it's because they make up the spindle fibers. These little barrels, these centrioles are found on one end of the cell in the cytoplasm, but during mitosis, they copied themselves, and we have a pair at each end of the cell. That's where spindle fibers will originate. They'll spread out in both directions. Some will attach to the chromosomes, some will just push or pull the cytoplasm apart. So they look like this. There's usually, they come in a pair at a 90-degree angle to each other, and then they're going to replicate themselves before mitosis starts. We have the same type of arrangement in the centrioles as we do in the cilia and the flagella too. So now something else we see. Cells need to be able to absorb materials, nutrients, oxygen, water, different proteins, different molecules. In the small intestine, we have a tremendous area for absorbing nutrients. All those simple columnar cells are designed to absorb nutrients. Well, to enhance that, a cell will extend the shape of its membrane to form what are called villi. Villi, the term villi means finger. And so we have villi. These things here are villi, these things sticking up. So the surface of the cell membrane goes up like this. These are villi. Now we have a much greater surface area. And in many cells, not only do we have villi, but we have microvilli projecting out on the side, on both sides like that. And that even increases the surface area for absorption even more. A typical small intestine, which is about 20 feet long and about as big around as your index finger, the total surface area encompassed by that 20-foot long tube, no bigger than your finger, if you stretch it out flat to its entire surface area, thanks to the folds of the villi and the microvilli, you could cover a structure the size of a tennis court. So it's this tremendous area for nutrient absorption, all packed together like this in our small intestine. And yet, if you stretch it out flat, one cell layer thick, which is what it all maintains as one cell layer thick, you'd have this huge area. So, we depend on this so that we can get all of our nutrients absorbed as it enters into the small intestine. So that gets us to the larger organelles inside the cell. In fact, the largest organelle, which is the nucleus. And the nucleus is where we find our DNA. It's usually in the center of the cell somewhere. Most cells only have one nucleus. Some are multinucleate. The skeletal muscle, striated muscle, voluntary muscle, is multinucleate because it was made up of lots of smaller cells joined together when we were in utero. It doesn't matter that there are many nuclei in skeletal muscle cells because skeletal muscle cells don't reproduce. They never make new ones. So they are multinucleate, but they are stuck in the cell cycle so that they can never get into mitosis. So, but if you have a cell with a nucleus, and only the red blood cells don't have a nucleus. Red blood cells had a nucleus, but they kick it out in one of the last steps of formation before they're released into the blood supply. They kick their nucleus out so there's more room for hemoglobin. So they're the only cells in our bodies that don't have a nucleus. Anyway, nucleus, site of the DNA location. We keep our DNA in the nucleus. That's our genetic information. The genetic information in each nucleus is exactly the same in every cell, regardless of what type it is, whether it's a liver cell, skin cell, bone cell, muscle cell, it doesn't matter. The DNA is exactly the same in each case. Only the information on how to be a liver cell is turned on for cells in the liver, or how to be a bone cell is turned on for a bone cell. Everything else is there, but it's just not turned on. It's not being used. It's like a cookbook. If you're using a cookbook and you go through and you find a recipe in how to, I don't know, make a meatloaf, now that cookbook is probably going to have recipes in there for desserts and salads and all sorts of stuff like that, but you're not using it. It's there. somewhere in the back or in the front of it, but you're looking at meatloaf. I don't know why, but you're looking at meatloaf. You have instructions on how to make meatloaf, if whatever. You're not going to be flipping page. Oh, look, here's how to make, here's how to make a cake or a pie or a roll or something like that. Anything but meatloaf. But you're not going to use those. You're only going to use the instructions you need. And that's what the cells are like. Bone cells only use the information they need to be a bone cell, but all the other information is still there. We don't pick and choose our DNA selectively. When the cells are being formed, they get all the DNA. They just don't have to use most of it. Anyway, if you're a nucleus, you've got a couple parts to you. You've got a nuclear membrane or a nuclear envelope made up of the same material that makes up the cell membrane. So you have a nuclear membrane, and you have a structure inside called the nucleus. And the nucleus is where we generally make the RNA we use to put together the ribosomes. And then you have something filling in you see here in the picture, the dark stained area of the nucleus is the nucleus, and you have the nuclear membrane out here in the outside. And then you have something called chromatin. Chromatin is a mixture of DNA wrapped around a protein called histone with a little RNA thrown in to help stabilize it. And that's how we protect our DNA molecule. DNA molecules are about 6 feet long. They're very thin and they're very fragile. So to protect them, they wrap themselves around these proteins called histones, and they make a larger structure. So now when the DNA moves around, it's going to move around with this protein surrounding it for protection. And this combination of DNA and protein and a little RNA is called chromatin. And normally we don't see that in the nucleus. If we look at a nucleus, the only time we would see the chromatin is when it condenses and forms the chromosomes. Because normally DNA is all unwound, it's wrapped around the chromatin, but it's all unwound, it's invisible. You look at a nucleus, you don't see anything. It's only when the chromosomes condense, when the chromatin condenses, do we actually see where the DNA is located. We don't see the DNA, but we can see the chromosomes. Okay. So we have the nucleus. It's filled with cell goop, just like the cytoplasm is in the cell, only we call it nucleoplasm. It's about the same texture, mostly water, a little goopy material. And it's very similar to what's the cytoplasm is. The outer part of the nuclear membrane has openings in it called nuclear pores. Everything gets called nuclear something here. So these nuclear pores are continuous with the rough endoplasmic reticulum, because we're going to make RNA. Remember what RNA is. RNA is usually made as a copy of a segment on the DNA. When we copy a gene, remember, a gene is nothing more than the physical location of a set of instructions on how to make a protein. When we copy a gene onto messenger RNA, we send it out to the ribosomes through these openings in the cell membrane called the nuclear pores. So we have to have these holes in the nucleus, and they lead directly into the rough ER. The opening leaving the nucleus itself, the rough ER is butted right up against it. There's an opening in the rough ER, so you go right from the nucleus. If you're a messenger RNA, you go right from the nucleus through the membrane right into the rough ER. And so those openings are called the nuclear pores. And we also see we talk about the skeleton of the cells. There's a layer of proteins wrapped around the nuclear membrane to give the nucleus its shape. And this is what this stuff looks like. Up here, get my little marker out here. Okay, here we see our nucleus right there. These little yellow things are the nuclear pores. And you can see how there's one right there that goes right from the nucleus into the rough ER. This is the rough ER right here. Now this is the nucleolus here where we make RNA. That's an A for ribosomes. All this is chromatin in here. And if we take a look at the actual microphotographs, you can see all these little circular dots here are nuclear pores. Here's a close-up of them, what they look like. There's the opening in the middle, but they have these little areas around. I mean, it's covered with pores. So we have, we have this complex which surrounds the opening to protect it. And then the lamina, the protein that gives the nucleus its shape down here, it's like a mesh woven back and forth, holding it in shape. So okay, let's get out of this and go to the next one. Now, the chromatin, as I said, is a mixture of DNA and a little RNA and a lot of protein. It's about 30% DNA, 60% histones, and 10% RNA. The DNA is wrapped around the protein. It looks very much like a candy cane. And so by wrapping itself around the protein, it provides some strength and stability to this very, very fragile molecule. Because again, it's microscopically thin, and it's a double helix, it's twisted, and it's about 6 feet long. But by wrapping itself around the histone protein, we protect it. We allow the DNA to be moved, to condense, to be dragged in chromosome form to opposite ends of a cell and not have it damaged. So, because all a chromosome is the condensed chromatin. Nothing has changed. The chromosome is nothing but the chromatin condensed. Now we have 23 pairs of chromosomes. Our parents each gave us a set of 23. 23 maternal chromosomes, 23 paternal chromosomes. That's 46 altogether. So we got 2 chromosome 1s, we got 2 chromosome 2s, we got 2 chromosome 3s, all the way out to the 23rd pair of chromosomes. And each chromosome is going to be made up of DNA wrapped around chromatin. Each chromosome is going to be different. Each pair is going to be different. For example, if we see they're going to have similar properties though. If you have here, for example, this is a chromosome 12. I'm just making this number. I mean, I don't know what's on chromosome 12 off the top of my head, but there's a chromosome 12 from the father and there's a chromosome 12 from the mother. And we may have a gene here and here for eye color. This may be the dominant gene. This may be the recessive gene. If this is blue eyes, and this is for brown eyes, the dominant always wins. If we have the same chromosome and we have the same gene location here, and we have blue eyes, the gene for blue eyes on both chromosomes, then blue eyes wins. Each pair of chromosomes are going to usually have the matching gene or the the dominant or the recessive gene or the pair. They can be dominant genes, they can be recessive genes, they can be a mix. But usually what will happen is that, whatever the chromosome number is, its counterpart, the male chromosome and the female chromosome, you really can't tell them apart unless you get deep down into the makeup of the DNA here. But you would have the genes that control the traits are generally going to be found in the same spots on the matching chromosomes. I probably told you more than you need to know on this. Okay, now, so we get 23 pairs of chromosomes in all of our cells except our sex cells. And when we were developing in utero, we had received 23 chromosomes from our father, and we received 23 chromosomes from our mother, giving us the full complement we need. So we have the same traits, and we have the same genes for hair color, eye color, skin color, whatever. The traits then depend on how the gene's going to be a dominant or a recessive gene. Is the trait going to be, as they say, expressed, or is it going to be hidden? So now, and this is what our chromatin looks like. And I say it looks like a candy cane. It's sort of a little bit of a stretch, but you can see the DNA is wrapped, is in red there. It really isn't red, but it's wrapped around the blue histone, which it really isn't blue, and it does sort of look like a candy cane. And by having this type of structure, we can condense this, we can coil it together, and we can get our chromosomes. Again, the chromosomes are nothing more than the condensed chromatin. That's all they are. Now, there are certain traits that always show up on certain chromosomes. And off the top of my head, I don't remember which ones they are, but if we get some time to talk about genetics in pregnancy, we'll, I'll bring out some of that for you. But there are certain traits that always show up in the same locations. So and we're getting them, the amazing thing is we always get them matched up. Okay, let's go on. Now, if you're a cell, you don't operate randomly. You don't operate by yourself. You operate in a collection of other cells called tissues, and you run on a cycle. You have what's known as the cell cycle. And the cell cycle is dominated by interphase. That interphase is what a cell does when it's living, when it's doing cell things. When it's making proteins, when it's killing bacteria, when it's, whatever it's doing, the cell life. The cell cycle represents 99.99% or more of cellular time, of the cellular activity. If a cell lives for 3 days, it's going to spend 99.99% of its time in interphase. Cell division, mitosis, and cell division represent probably 1/100th of the time of the cell cycle. Cells spend very little time in mitosis and cytokinesis because they're very fragile at that point. They are vulnerable at that point. And so they go through the whole process, they replicate their DNA, and then they go and copy themselves very quickly so they can protect themselves. Because if a cell spent, well, I'm going to go into mitosis now, so for the next 3 days, I'm going to be busy copying myself and replicating. And I won't be able to fight off any infections. I won't be able to fight off any pathogens or anything like that. I won't be able to do anything because I'm busy copying myself. Well, that's not what a cell does. A cell spends seconds, if that. A typical cell may live for a week, and then its last 5 seconds is spent in mitosis and cell division. because it doesn't want to waste a lot of time, being vulnerable and being unfed while it's producing two daughter cells. So interphase, what a cell does when it's busy being a cell, and then mitosis and cell division are a very, very brief form. Now, during interphase, what are we doing? Well, we're doing all the cell things we do. We go after bacteria, we destroy viruses, we destroy broken fragments of cells, we destroy any kind of pathogen, we protect the cell. We're going to be busy making proteins and cholesterol and hormones, and we're going to be growing and getting stronger and do whatever we are supposed to be doing. Absorbing nutrients, for example, or contract. Well, we're not going to be contracting anything because those cells don't. They're going to be contracting in the cell cycle. They're going to be busy in interphase and they never get out of interphase. So, interphase actually has three subsets: the G1 stage, or the gap 1 stage, growth and metabolism. Whatever kind of cell you are, if you're in the G1 phase, you are growing and you have a high metabolic rate. You're doing all sorts of neat cell things. Well, muscle cells never get out of G1. They never grow up, if you will. But other cells are busy. They grow rapidly, and then once they reach any adult size, then they're going to keep on doing what they've been doing. And they are going to run through this G1 phase as a subset, probably most of their cycle, their life cycle. Now, cells that don't divide don't ever get out of G1. We actually say they are in G sub 0. Muscle cells don't divide. We've got all the muscle cells we're ever going to get. All they can do is die, but we can't grow new ones. When we exercise and work out and our muscles get bigger, we're not growing new muscle cells, we're just making the ones we have larger and more powerful. Nerve cells generally don't undergo mitosis either. We are pretty much stuck with the nerve cells that we have now, what we were born with. We may lose some over time. In fact, nerve cells do wear out, just like muscle cells wear out, and we lose some. But we usually don't grow new ones. So both nerve cells and muscle cells are essentially stuck in the G sub 0 stage of the cell cycle. Everything else is going to go through G1. It'll spend most of its time in G1. There's another subset here called the S stage, or synthesis stage, where we are going to replicate our DNA prior to mitosis. Because if we're going to form two daughter cells, each daughter cell's got to have the exact same set of DNA that the parent cell had. Got to be perfect copy. So we got to replicate our DNA. We got to make a perfect copy of our DNA, but we can't have two sets of DNA in the cell for very long because they would have competing sets of instructions. So we have a very brief S stage, synthesis stage. We take the time, though, to make sure that there are no mistakes in the DNA, because mistakes happen. Mistakes happen all the time. We have ways to fix that, but mistakes can happen, or we can detect damage. But if our DNA is fine and we make a perfect copy, we're good to go. And then the last stage before mitosis is called the G2 stage. In G2, we are completing the actions necessary for mitosis to occur, enzymes that we need for mitosis, any other chemicals we need, any other molecules we need are going to be present. And we're checking again to make sure that our DNA is perfectly copied. And then if everything has a green light, then we're going to go right into mitosis. And the cell cycle looks something like this. We see that we start here. We start right here as a brand new daughter cell at G sub 1. This is the first stage of interphase. Now, interphase is going to run along the outside here all the way from G1 to the start of mitosis. Now, so the G1 phase will continue. Interphase will continue the whole time the cell is in this cycle, except for the mitosis phase. The actual time spent in mitosis is probably equivalent to just this line here. It's not this pie-shaped segment that we see here. That's a lot of time wasted. The actual time spent in mitosis is probably just this one line. It's over in seconds. We also probably don't spend this huge amount of time in the DNA replication stage either, because we don't want to have, a cell doesn't want to have another set of DNA in its nucleus or where its nucleus was for 2 or 3 or 4 days, giving conflicting instructions to the cell. So again, that's probably a pretty short stage too. But up until the point where we get ready to go into mitosis, we're going to be doing all sorts of cell activities. So let me clear this all for you. So here we see the G1 phase. We start over here, right as soon as we're a brand new daughter cell. We start here, the interphase starts there and goes all the way to the beginning of mitosis. This first part is the G1 phase. The next part is the S phase. The next part after that is the G2 phase. And then we have mitosis followed by cytokinesis. And again, the actual amount of time spent in this is much shorter than this diagram shows. Okay. Now, to copy our DNA, we have what we call a perfect copy. We have to make a perfect copy of our DNA. Let me back up here. And the process is known as semi-conservative replication. Let me get this out of the way here. The process is known as semi-conservative replication. And essentially what we do is we take one strand of DNA, take the double strand of DNA, split it in half, and use each half of the DNA strand as the model to make 2 new strands. So if you have your strand here, you take your strand and split it apart. And so, for example, here is guanine that was attached to cytosine. Now, guanine pairs to cytosine and thymine pairs to adenine. And so our strand splits apart. And now you start building in 2 new strands here and here, so that this is adenine, this is guanine, this is thymine here, and so on. Like you can see thymine and adenine. So here's adenine, there's thymine. You build 2 new strands that are the complement of the original strands. You split it in half. Now, what do you need to remember about this part here? Well, to start out with DNA replication, we have to split the strand in half. The first thing we do to split the strand in half is we use an enzyme known as helicase. Helicase opens up the double strand. Let me go back here and, oops, okay, now, there's our normal DNA strand. There goes the helicase across there. Helicase is an enzyme that breaks the hydrogen bonds that are holding the bases together. So the DNA strand has unwound, and we call where it's now exposed like that, we call it the replication fork. The strands have separated, and we're going to start building new DNA strands off the original. So this way, the term semi-conservative replication means we're using each original strand, the left side and the right side, as the basis to make two new strands. They're a template, they're a model. So with semi-conservative replication, we start building two new strands off the old one. We use another enzyme to make this work, and we call this, this enzyme is called DNA polymerase. It starts bringing the nucleotides together that are the complement. That's what this purple thing is. It looks like a lifesaver wrapped around. This is DNA polymerase here. And it moves along the strand, and it sees what the base is on the strand, and it builds the new strand in a complementary pattern. So if the original strand had adenine, the new strand has to have thymine. Thymine is a complement to adenine. If the old strand had cytosine, the new strand has to have guanine, because guanine is a complement to cytosine. And so we build our strands using the DNA polymerase. Now, we use one more enzyme. First of all, we've seen helicase opens it up. DNA polymerase builds the new strands. We don't just run from all the way at one end to the other. You know, that thing is 6 feet long. It would take days for that enzyme to run, that polymerase to run down the strand. It would take days for the helicase to open everything up and to run the enzyme, the polymerase, down the strand. We do it in segments. We do it in multiple places. And so we're putting together these, we're making all these segments of DNA, of copied DNA. So we use a third enzyme called ligase, which is the DNA superglue. Ligase glues these sections together. We make them, we open it up in segments, we make the new DNA in segments, then we glue the segments back together again. And we can make 10 billion copies, 10 billion pairings, you know, adenine to guanine, cytosine to thymine, and so on. We can make 10 billion of those in about 2 hours. We can make 2 new strands in about 2 hours with about 10 billion pairings, with an error rate of about 1 in a billion. That's pretty good. So we build, for every original strand of DNA, we can make 2 new strands, because we got to have one, we got to have the strand, each daughter cell has to have a complete set. And their DNA, the daughter cell DNA, is going to be half original DNA from the maternal cell and half new DNA. You can't tell the difference. You can't tell it apart. It looks exactly the same. It acts exactly the same. It is the same. It just half is the original and half is the new copy. Let me turn the sound on here for us. Let's see if this works for us now.
Copying DNA, a process called In principle, copying DNA, a process called DNA replication, is very simple. The 2 complementary DNA strands separate, and because each nucleotide can only pair with its complement, A with T and C with G, each strand can be used as a template to build a new complementary strand, producing 2 DNA molecules. In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we have untwisted the double helix. DNA replication begins at specific sites called origins of replication. Proteins attach here and separate the DNA strands, forming replication bubbles which grow in both directions. Enzymes called DNA polymerases move along the template DNA strands and catalyze the elongation of new strands. Eventually, all the replication bubbles merge, yielding two identical DNA molecules.
So you see how the helicase opens up the DNA, the original DNA strand, into two separate strands. Then we use the DNA polymerase to build the new strands in multiple locations. And then we use the ligase to glue it back together. And we end up with two brand new strands, one with the original strand, the template, one with the new strand we just built. And all we have to do is match the adenines to the thymines and the guanines to the cytosines, and we're complementary. And we end up producing two brand new but identical strands. This is how we copy our DNA in the S stage here. Now, there are always a chance of an error occurring, and they will occur. And we have enzymes that slide up and down the DNA after it's been copied like this to make sure that there are no mistakes. If there's a mistake, the enzyme can cut out the error and replace the incorrect nucleotide with the proper one, because mistakes will happen. Sometimes you'll get a guanine paired to an adenine, or a thymine to another thymine, or a cytosine to an adenine. It can happen. When you're making 10 billion pairings, it's not difficult to make an error. The normal error rate before it gets corrected is usually about 1 in a million. But after we fix it, after these enzymes go through here and fix the problem, the error rate drops to something to the neighborhood of 1 in a billion. So the error rate pretty much goes, the error goes away. Now let's, okay, so that is the semi-conservative replication phase that we get. This is how we copy our DNA. This is how we make a perfect copy of our DNA. And that leads us into the last step of the cell cycle. Well, we have the G2 phase where we get everything ready. But the last step of the cell cycle is mitosis and cell division. Muscle cells don't divide. Skeletal muscle doesn't divide. Cardiac muscles don't divide. Nerve cells don't divide. If the cell gets damaged, we have scar tissue in place of it. In skeletal muscle, if we damage our voluntary muscles, we have scarring. In the heart, if there's damage to the heart from a heart attack, we have scarring. Scar tissue doesn't do anything except fill in the place. Scar tissue is not contractile, and it is not going to send nerve impulses. Same thing applies with nerve tissue. Now, the mitotic phase, mitosis phase or mitotic phase, the M stage of the cell cycle is mitosis followed by cytokinesis. And we know cytokinesis starts in anaphase. So, we'll just leave it like it is, I say here. Now, we control the rate of cell division. We don't want to have uncontrolled cell growth. Uncontrolled cell growth is cancer. We don't want to divide when we don't need to. Cells only divide when they are wearing out. Cells only undergo mitosis and cell division when they're starting to wear out and they need to be replaced by new cells. Cells grow and replicate to meet the demands of the body. And we don't get unnecessary growth. Cells also maintain what we call contact inhibition so they don't overgrow each other. This, you've seen this before from lab, but this shows you just how rapid the mitosis stage is. Prophase, metaphase, anaphase, and telophase are done in seconds. You can see just how short a time frame this is. When particularly, I like this one here because you go from anaphase to telophase so rapidly. Anaphase is the shortest stage of any of the four stages of mitosis. So we have prophase, metaphase, anaphase, and telophase. We've talked about these in lab. Get a little more detail on them here. Now
Cell division consists of two processes, mitosis and cytokinesis. Mitosis, division of the nucleus and its chromosomes, is divided into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. Mitosis is followed by cytokinesis, when the cytoplasm splits to form 2 separate daughter cells. The cytoplasm of a cell in late interphase contains 2 centrosomes, each of which may contain a pair of centrioles. In the nucleus, the chromosomes have been replicated during S phase, but are still dispersed in the form of chromatin. 1 or more nucleoli are present. During prophase, the nucleoli disappear and chromatin fibers coil up to become discrete chromosomes. Each chromosome consists of 2 identical sister chromatids joined at the centromere. Microtubules grow out from the centrosomes, initiating formation of the mitotic spindle. During prometaphase, the nuclear envelope breaks into fragments. Some of the spindle fibers reach the chromosomes and attach to kinetochores, structures made of proteins and specific sections of DNA at the centromeres. Non-kinetochore microtubules overlap with those coming from the opposite pole. At metaphase, the mitotic spindle is fully formed, and the microtubules attached to kinetochores move the chromosomes to the metaphase plate, an imaginary plane equidistant from the poles. Anaphase begins when the 2 centromeres of each chromosome come apart, separating the sister chromatids. Once separate, each sister chromatid is considered a full-fledged daughter chromosome. Motor proteins of the kinetochores walk the daughter chromosomes along the spindle microtubules toward opposite poles, and the microtubules shorten. At the same time, the spindle microtubules not attached to chromosomes lengthen, pushing the 2 poles farther apart and elongating the cell. During telophase, nuclear envelopes form around the identical sets of chromosomes at the 2 poles of the cell. The chromosomes uncoil and nucleoli appear in the 2 new nuclei. Meanwhile, cytokinesis begins, splitting the cytoplasm and separating the 2 daughter cells. In animal cells, cytokinesis begins with the formation of a cleavage furrow. At the site of the furrow, a ring of microfilaments contracts, much like the pulling of drawstrings. The cell is pinched in 2, creating 2 identical daughter cells. In plant cells, cytokinesis begins when vesicles containing cell wall material collect in the middle of the cell. The vesicles fuse, forming a large sac called the cell plate. The cell plate grows outward until its membrane fuses with the plasma membrane, separating the 2 daughter cells. The cell plate's contents join the parental cell wall. The result is 2 daughter cells, each bounded by its own continuous plasma membrane and cell wall.
we don't have a cell wall. We just have our cell membrane. And so we see the different stages here. Something else, prometaphase is a term that is rarely used when we talk about the stages of mitosis. So don't get hung up on that prometaphase stage. We talk about prophase, metaphase, anaphase, and telophase as the 4 stages of mitosis. We generally don't throw in prometaphase, at least in A and P we don't. Anyway, you can see everything that has to happen, and it's a pretty complicated process, and yet we end up, it works pretty well every time for us. So prophase, we know what happens in prophase. The nuclear membrane disappears. the chromosomes are starting to condense. We may start seeing the spindle fibers forming from the centrioles at opposite ends of the cell. The chromosomes have condensed and usually pair up together. They are held together by a structure known as the centromere, sort of like they're temporarily glued together, like the two chromosome 1s and two chromosome 2s, and so on. The original set and the duplicate set, and we start seeing them beginning to move towards the midline of the cell. The spindle fibers may attach to them and start dragging them to the midline. We may see the spindle fibers, as they're forming, start pushing the cytoplasm a little bit in here. And our cell starts to stretch out just a little bit. Okay. Metaphase, the most obvious stage, the one that is the easiest to spot, where the chromosomes have lined up across the equator of the cell. We call it the metaphase plate. And the only reason they do this is to guarantee that we're going to get an equal distribution of 23 pairs of chromosomes going to the left side and 23 pairs going to the right side when we get the two daughter cells. We want to make sure there is an even distribution of all the chromosomes. Anaphase, the shortest stage. It's also the stage where cytokinesis starts. The chromosomes are being pulled apart so that you get an equal distribution on both sides. You want to get both daughter cells having 23 pairs of chromosomes. And so you have your pair split in half. And so your chromosome 1s are going to go to opposite side, your chromosome 2s are going to go to opposite sides. And since you have two sets of DNA, you're going to end up with your 23 pairs. And then, this whole process is very, very quick. Anaphase is very brief. Telophase, the chromosomes unwind, the nuclear membrane reforms. Cytokinesis terminates in here with the two daughter cells forming, and again, the nucleus is visible. The nuclear membrane reforms, and the chromosomes disappear. They're still there. They're just unwound. And then telophase. Again, this is where we start seeing the separation. Where we see cytokinesis is complete. Cytokinesis starts in anaphase. It finishes in telophase. And where telophase ends and cytokinesis is complete is very problematic. It's really hard to look at it and say what's going on where. This is what cytokinesis looks like from the outside of the cell. This cleavage furrow, this line, is where you have a ring of actin. Actin is one of those microtubules, microfilaments. It's a contractile protein. We see actin again when we're looking at skeletal muscle. But actin can contract, and it will squeeze the cytoplasm. Excuse me. It will squeeze the cytoplasm and pinch it apart in here. And this process will continue as the actin gets tighter and tighter and tighter here until the cytoplasm is totally pinched apart and you have two brand new separate daughter cells. Now we have a series of control mechanisms here. We have go and stop signals all the time. This isn't going to just run randomly whenever it feels like it. These go and stop signals tell the cell when it's time to divide and when it shouldn't. The go signal is when the cell's gotten too big, or when the cell, the demands of the cell are greater than it can provide for itself. When we're talking about the surface to volume ratio, very large cells have a problem feeding themselves because they have a great big volume and a little itty bitty surface on the outside. They're really big cells. The surface area outside is nowhere big enough to satisfy the volume of the cell. So that's why we are not made up of just like a half a dozen really big cells. We're made up of billions of cells. Little cells with a surface area that matches the volume work just fine. So we would grow, we would undergo mitosis if the cell started to grow bigger than it could provide its own nutrients for. It would also grow when it's exposed to certain hormones, like growth hormone. You know, when we hit puberty, we start growing to our adult stature, and we release lots of growth hormone to stimulate bone growth and muscle enlargement. We release lots of thyroid hormone to provide energy for this activity. Usually, and this is all driven by the sex hormones. You know, as we hit our adult stature, the driver for that is going to be the release of testosterone and estrogen. We're going to hit our adult stature, and we're going to have our secondary sex characteristics appear. And you know, all of these factors are going to come into play. We can have occurrences of excessive growth, inappropriate excessive growth, if we release too much of some of these hormones. You know, excessive amounts of growth hormone can result in a condition known as gigantism. Too little growth hormone can result in a condition known as pituitary dwarfism. And we'll talk all about that in chapter 6 on bones. Now, those are all the go signals. Now, the stop signals that tell us when to not undergo mitosis is if the cells are too crowded. The cells are too crowded. Cells have contact inhibition. Just like we don't like being crowded. We don't like to have people, you know, standing up next to us, leaning over our shoulder and things like that. We are very uncomfortable when that happens. Consider an elevator. When one person goes into an elevator, they usually stand in the middle of the elevator near the doors. If a second person comes in, then without saying anything, most people end up going into the corners and standing in the corners because we don't like to have people too close to us. And if a third person were to get on, they got to stand in the middle between the two of them, but up in front by the door. That's just the rules. We all know that. We've all seen that. You know, it's not really the rules. You know what I'm talking about. We do that subconsciously. We don't think about it because we don't like to be too close to other people. Most of us don't like people anyway. Cells are the same way. That's their version of contact inhibition, is to stop undergoing cell division and mitosis if they get too crowded. If they bump up against another cell, they're not going to grow anymore for a while. Contact inhibition stops that. Cancer cells don't have contact inhibition. Cancer cells can grow on top of other cells, so they don't care. But a normal healthy cell will not undergo mitosis. And so we get a great big stop signal if the cell starts to get crowded. Another type of control mechanism we have here for our cell cycle are these two specific proteins. They are called cyclins and cyclin-dependent kinases. Kinases are enzymes. Cyclins are proteins, or we call them a regulatory protein. We keep manufacturing them during the process of interphase, and they are inactive until we start making these cyclin-dependent kinases. Like in our bodies, we find that most of our special activities, like clotting blood, for example, depend on a multi-step process. You know, we just don't say, "Oh, we got to cut. Let's start clotting." Well, before we can have a clot form, when we cut ourselves, we have to turn on one protein which activates a second protein, which activates a third protein, which activates the clotting protein. We don't want to start clotting accidentally, which has nothing to do with mitosis other than the fact that it's the same principle of we have to produce one protein to turn on a second protein, which regulates when the cell cycle is going to run and when it's not going to run. When are we going to go to mitosis and when we're not going to go to mitosis? So the cyclins build up during interphase, and these enzymes, the cyclin-dependent kinases, that means these kinases, these enzymes depend on the presence of cyclin. And when the kinases bind to cyclin, the cyclin becomes activated. And cyclin, this complex of cyclin and the cyclin-dependent kinase, the enzyme, when cyclin becomes activated, then we can go into mitosis. We build these cyclins all during the process of interphase. We build these enzymes, these kinases that are the cyclin-dependent kinases. We build them the whole time that we are in interphase. If they come together and if they activate the cyclin, then and only then can we start mitosis. So not only do we don't grow if we're crowding other cells, but we also have to have these cyclins turned on. And we start all over again. We destroy them all. As soon as at the end of mitosis, they're all destroyed. So we start fresh every time. There's no leftovers, if you will. Okay. Checkpoints. We got to have checkpoints. We depend on multiple places to make sure everything's being done right. These checkpoints tell us when everything is ready and that the DNA is copied properly. We have to have everything lined up ready. The G1 checkpoint is the most important checkpoint. The G1 checkpoint is at the end of the G1 stage. And here, what we're doing is we are checking to make certain that the DNA, our DNA, is in perfect condition. We haven't tried to copy it yet. Remember, at the end of the G1, we're getting ready to go into the S stage. If there's a mistake in our DNA, if our DNA has been damaged by something, sunlight, ultraviolet radiation, chemicals, if there's a mistake in our DNA, we're not going to go into the S stage. If there's a mistake there, because we examine the DNA before we ever try to copy. Because if we copied it with a mistake, then we would pass the mistake on to the daughter cells. We don't want to do that. So we have to check the DNA to make sure it's perfect. And we do that at the end of the G1 stage. That is the most important checkpoint we have. We have a second checkpoint at G2 to make certain that the copied DNA is good. And there's another checkpoint in mitosis at the metaphase point where we check to make sure that everything is being separated properly. So we have three big checkpoints here: the G1, the G2, and the M checkpoint. But the G1 is the most important. So, because we're checking for damage. The G1 checks to see if the DNA, if our DNA is damaged. If it's damaged, we have two choices. If we have damaged DNA, we have two choices. Self-destruction, called apoptosis, where the cell will release its own digestive enzymes and destroy itself. Apoptosis, or fix it. If we can fix it, we will. If we can't fix it, then the cell destroys itself. Those are the two choices. Well, there's a third choice where the cell goes into what we call senescence, where it just becomes in suspended animation, if you will. It doesn't do anything. So you're either going to initiate self-destruction, or you're going to fix it, or you're going to go off into a corner and sit there and never do anything again. So either way, why not just destroy yourself? Anyway, so that's the first checkpoint. The second checkpoint is to make sure that we didn't copy any errors in the DNA. That's the G2 checkpoint. And then in the M checkpoint, we're going to check to make sure that everybody's getting the right DNA they're supposed to get. So we got 3 places where we're going to stop this process, and it will stop the process cold. It will stop it right in its tracks. And if there's something wrong with the DNA, it will stop everything. It will not let anything slide through. Even the slightest mistake will cause it to be stopped and either repaired or destroyed, and take the cell with it. Okay, this is a good place to stop here on mutations. We'll pick up with mutations on Monday and move on into more talk on how we produce, actually, how we use our DNA to produce protein. So we'll stop here at this point. Questions on anything? Okay, if you have any questions on the test of a general nature, better throw it out there. Feel free to ask at any time. Otherwise, I will see you all on Monday.